Hello, friends, and welcome back to the channel. It's always beautiful as we have you here, and we continue our discourses in science. For today, remember we've been talking about DNA sequencing, which talks about the process by which we are able to understand the alignment and the arrangement of different nucleotides within a DNA molecule, and that's what DNA sequencing is about. And it has applications in many areas of science. Now, for today, we are going to look at the process, step by step process by which we do DNA sequencing, and it starts with DNA extraction. And what's DNA extraction? DNA extraction is one of the ways or the methods by which we are able to bring out, remove DNA that is present either within a living cell or even a cell that is no more living, or even from a viral particle. Any cell or viral particle or any living thing, or like a virus that has DNA, we are able to harvest that DNA. We're able to collect that DNA from out of the cell, because that's the very, very, very first step when we come to DNA sequencing, the DNA extraction, the removal of the DNA itself from within the living cell. It could be an animal cell, a plant cell, a fungi cell, even a virus. From any of these things, we could remove our DNA. Now, what's the process? What does the process entail, and what does the process involve? There are many methods. One of the methods is the boiling method, which is one of the oldest methods of doing DNA extraction. And in the boiling method, you have a method where you have your sample, whether it's a plant cell, a bacterial cell, an animal cell. You have those samples and you have them immersed in distilled water. When you have the samples immersed in distilled water, then you have the whole mixture heated. It's heated at between 95 to 100 degrees Celsius for a period of time, 10 to 15 minutes. And when this is done, it's believed that the cell is broken up. And remember that there are many, cellular constituents. So those cellular constituents are broken up, and then your DNA is is extracted from within this cell. Now, this process is used in low resource settings, places where there is not enough resources, and this process is used, but it's also functional. Although the trade-off, the problem with the process is that sometimes there's a high amount of impurity present in the DNA samples. But it's important to know that this process exists and people do use this process for their DNA extraction. Now, also, the second process is the use of commercial kits. There are a lot of commercial kits that have been designed by companies, like Kyajin, like Zymo. All these companies have designed these kits, and these kits are effective. They are quite expensive, especially for we researchers in Africa, but they are very effective, and within an hour, you are able to extract your DNA. And the process works in quite a simple way. The DNA, we know that the DNA has the capacity to bind to magnetic beads or some other silica membranes, magnetic beads and silica membranes. So the process is simple. There's usually a lysis buffer. You have these processes being done where you have a lysis buffer. And when you have this lysis buffer, which is part of the kit, this lysis buffer helps to break down the cell and break down some of the other components within the cell, the proteins and the carbohydrates, and then release your DNA. But it's all in a solution. And then when you have this DNA in your solution, you also want to have your silica gel, silica membrane, or your magnetic beads to which this DNA are able to bind. And then you centrifuge, at very high revolutions per minute. And after centrifuging, the supernatant, that's the upper part of your centrifuge, you collect it, and that contains when you're able to collect your supernatant, you now have your DNA, which has attached to your silica membrane. So when you have that, you now have the DNA, and then you have it in the form where you can check for its purity, check how much of it that you have, the quantity and the purity. You also have the CTAB method. And the CTAB method is a more chemistry inclined method, which not many life scientists use this method, but it's also for learning purposes. It's good that we know. And there's a particular chemical called cetyl trimethyl ammonium bromide, which we call CTAB. And this chemical is used, and when you grind samples, usually it's used for plant samples and fungal samples. When you grind these samples and then you mix it with the solution of this CTAB buffer, this solution helps to break open the cells, and separate the DNA from polysaccharides from proteins and allows the DNA. Then sometimes or during the process they add we add chloroform to also bind to proteins, carbohydrates, and other components of the cell so that we have pure DNA coming out of that mixture. And in order to get that mixture, the DNA is carefully collected using cold isopropanol that is used to precipitate the DNA out of the solution. Then the precipitated DNA is washed and is dried, resuspended, and used for the analysis. So this is possible and this is done in some places. But usually the kit method is the most common method because it's faster, it doesn't take much time, and then it gives higher quality of DNA. And you know, when you're talking of quality of DNA, you're talking of DNA with less impurity. Now, how do we check for the quality of DNA? The quality of DNA can be checked with quite a number of methods. The simpler method that we use is the method of using your gel electrophoresis. When you do your gel electrophoresis, you are able to check for the bands of your DNA. Now, your gel electrophoresis is a process where you get to run your DNA through using agarose gel, and then you have a field, an electric field is produced about 1.5 volts, and then you have the agarose gel, and then the DNA is loaded at one part of the tank, and is then along with the DNA you load in a coloring material, either ethidium bromide or your SYBR green dye. And the idea is for visualization. So when this is done, and then you later have the process that starts, and then you watch the bands of DNA. The DNA is really clear. If it's really pure, sorry, you see very clear bands, single clear bands, which is an indication that you have very pure DNA that can then be used for other analysis. We also have the nano drop spectrophotometry, which is another method that you use to check for the purity of DNA. And that method uses absorbance of light, spectrophotometry, where the DNA sample is, subjected to different kinds of light, and then the absorbance that they give off tells you the exact quality of the DNA that you have in the sample. So these are the methods we use, and when you run them in the laboratory, you tend to understand them even better. But extracting DNA is important because it's the first process. We extract our DNA, we do our library, we do our fragmentation, and then we have do the sequencing, and then we do the blasting. These are all the processes, and we'll be discussing them one by one by one. Thank you for listening, and do have a beautiful day. Cheers.